Hello everyone. In the fourth International Eurasian Conference on Biological and Chemical Science. Thank you for the opportunity given by organizers to present my work as a video presentation. My name is Lina and I am a master student in Bolu Abant İzzet Baysal University in Bolu, Turkey. Today I would like to share with you a part of my work which is detection and characterization of antimicrobial peptides of Lactobacillus plantarum B14 isolated from infant feces. And this work is supervised by Prof. Dr. Mehmet Öztürk. The plan of my presentation today. I will start with a small introduction of the context of my work, then I will talk about the objectives, followed by materials and method used in this work, then the result and discussion, and finally the conclusion. Large amounts of antibiotics are used to treat or prevent some types of bacterial infection. Infectious diseases have become the most important problem as a result of the presence of pathogenic bacteria that have multi-drug resistance. Antimicrobial peptides synthesized by certain bacteria that can be used as alternatives to traditional antibiotics. Lactic acid bacteria are probiotics that gained importance because they are considered as safe, found normally in the gastrointestinal tract of both human and animals, and they have the ability to produce antimicrobial peptides. Alternative solutions against antibiotic-resistant pathogens are urgently needed. The aim of this study is to isolate and characterize antimicrobial peptides produced by Lactobacillus plantarum B14 strain, isolated from feces of an infant. In this study, antimicrobial peptides of Lactobacillus plantarum was analyzed by applying different methods. The PCR reaction to detect the bacteriocin producing gene and cloning using p-blue script vector to detect the exact nucleotide sequence of bacteriocin producing gene, and then diffusion Kirby-Bauer method to test the bacteriocin activity against pathogenic bacteria. Materials used in this study is Lactobacillus plantarum B14 strain isolated from infant feces was obtained from BIO Biology Department in the stock culture stored at -80. Also primers were designed for PCR reaction supplied by oligomer. P-blue script vector and XL1 blue competent cell were prepared for cloning. And also for diffusion Kirby-Bauer method, 5 different pathogenic bacteria were obtained from microbiology lab of BIO, which them Listeria monocytogenes, Staphylococcus aureus, and pathogenic Escherichia coli, Salmonella typhimurium, Yersinia enterocolitica. Now, in the method, PCR reaction was used to detect antimicrobial peptides in Lactobacillus plantarum B14 using special designed primers by oligomer. Genomic DNA was isolated by GeneJet genomic DNA purification kit by thermoscientific. The table shows primers for both plantaricin S and plantaricin W. PCR condition was determined according to the instructions of the amplicon enzyme used in this reaction and the melting temperatures of the primers. In cloning step, as shown in the figure, first p-blue script was linearized by SEMA1 restriction enzyme. Then the PCR product was ligated using T4 DNA ligase enzyme. Then this recombinant DNA was transferred into XL1 blue competent cell of E. coli. Now for pathogenic test, diffusion Kirby-Bauer method was used to calculate the inhibition zone of antibacterial activity of Lactobacillus plantarum B14 in both cell-free supernatant and cell lysate against 5 different pathogenic bacteria. Cell-free supernatant and cell lysate was collected by centrifugation and loaded into blank discs, and these discs was applied on the plate with pathogenic bacteria. Now with the result and discussion, PCR product was purified by gene gel extraction kit by thermoscientific. As shown in the figure, M indicate the marker from Vivantis 1 kb DNA laser, and number 1 indicate the result from plantaricin S primer, and the number 2 show the result of plantaricin W primer. Now, the circular P-blue script plasmid DNA was isolated using the thermoscientific GeneJet plasmid mini-prep kit. We can see in the figure the circular plasmid size is 2000 kb, but then this plasmid was linearized by SIMA restriction enzyme and the size is increased to 3000 kb. In transformation to detect the insert of gene in a plasmid, plasmid isolation was performed by plasmid mini-prep kit. We can see in the figure, number 1 indicate the circular P-blue script plasmid, and number 2 and 3 indicate plasmid with a plan W insert result. Number 4 indicate plasmid with plan S insert result, and we can see the different incisors very clearly. For sequencing result, the Bacteriocin genes was sequencing by macrogene in Courier using universal primers, and the result was read by using bioedit program. And the nucleotide size for plantaricin W was 552 base pair. And here the nucleotide result for plantaricin S, and the size is 977 base pair. In silico analysis using Exposito, the following amino acid sequence were obtained. For plan W, frame number 2 is used with the start and stop codon. For plan S, frame 3 is used with the start and stop codon also. Then, after detecting the amino acid sequence, these amino acids compared using BLAST tool and NCBI. Then alignment was made using MegaX program. We can see the similarity in the phylogeneity is low with the other bacteria species. For plan S, we can see from the phylogeneity the similarity between the plan S result and the Enterococcus faecium is very high. Pathogenic test applying Kirby-Bauer method. The clear zone of inhibition around the disc loaded with cell lysate and cell-free supernatant indicates the activity of the bacteriocins against these pathogens. Here we can see the cell lysate from Lactobacillus plantarum shows activity against E. coli with inhibition zone of size 15 mm. And for the other result, cell-free supernatants from Lactobacillus plantarum shows activity against Listeria monocytogenesis with inhibition zone of 8 mm. And also cell lysate from Lactobacillus plantarum shows activity against Staphylococcus aureus with inhibition zone of a size 13 mm. To conclude, our results suggest that plantaricin S and plantaricin W peptides of Lactobacillus plantarum B14 may have antimicrobial activity against some pathogenic bacteria, but they need to be purified and further characterized for detection of this activity. And in the end, thank you for the attention.